All right, welcome back to our lectures. This is lecture number 1 for test two's material. Um, we will be discussing in this section the autonomic nervous system and how it relates to homeostasis. Okay, so let's do a little bit of an introduction here. If you've ever been driving your car and you've unexpectedly had to slam on the brakes to avoid hitting someone that maybe stopped too suddenly in front of you. After that event happens, do you remember how shaky you feel? Like maybe you feel a little sweaty and a little out of breath, but there's no physical exertion, you were just startled. Well, that feeling, along with your ability to hit the brakes so fast, is courtesy of a neurotransmitter called norepinephrine. And this is produced by neurons of the largely involuntary arm of the peripheral nervous system, the autonomic nervous system. Okay, this involuntary arm of the PNS is the autonomic nervous system, also known as the visceral motor division. You guys learned about the somatic motor division. Now this is the visceral motor division. And we've already discussed how the CNS maintains homeostasis. And I want you guys to remember out of that, it's maintained by the hypothalamus and the reticular formation of the brainstem. And much of these events are mediated by neurons in the ANS. Okay, and if you guys remember, we did discuss two different branches of the ANS, the sympathetic and the parasympathetic. If you want to look back on that, you'll go to 13.1. Chapter 13.1 shows you how they're related. A good way to remember is sympathetic is fight or flight, and parasympathetic is rest and digest. As with any other part of the nervous system, the ANS neurons communicate with one another at synapses, and these effects are mediated by neurotransmitters that bind to receptors on target cells. And the sympathetic and parasympathetic we're going to cover separately, but bear in mind that they work together constantly to maintain homeostasis. And then we will discuss at the end of this chapter how the ANS and the nervous system work together entirely to maintain homeostasis, sort of as a whole. So just as an overview, yes, we'll discuss an overview of the ANS. We'll discuss the sympathetic and the parasympathetic nervous systems separately, and then we will discuss how homeostasis, how the PNS operates to maintain homeostasis. Reviewing, you guys should remember this stuff. The autonomic nervous system, or ANS, is the involuntary arm of the PNS, the peripheral nervous system, also known as the visceral motor division. It is divided into two separate systems, the sympathetic and the parasympathetic nervous systems, and they constantly work together to maintain that homeostasis. These are what are going to oversee your most vital functions like heart rate, blood pressure, digestive system, urinary process, and these autonomously operate without any conscious thought. There's no thought put into these actions. So the ANS system oversees, many of those functions, and it is named for the fact that it can operate autonomously. And it performs these functions with a series of visceral reflex arcs, okay? These visceral reflex arcs are what are going to be crucial for the operation of the ANS systems. As you guys learned last time, a reflex arc is a series of events in which a sensory stimulus leads to a predictable motor response, okay? The basic reflex arc for the visceral reflex system, the basic arc for the visceral reflex arcs of the ANS are going to start with sensory signals from the viscera and the skin, okay, and these are sent by afferent sensory neurons to the brain or the spinal cord, where the stimuli are then integrated by the CNS, and then after they were integrated, the motor impulses from the CNS are going to be sent out through efferent motor neurons in cranial or spinal nerves, and these nerves will typically lead to ganglia in the PNS called autonomic ganglion. Okay, and then finally that autonomic ganglia sends the impulses through different efferent motor neurons to various target organs, where they will trigger that predictable motor response in the target cells. Here we have a great example. This starts, let's say this is the stomach, and it begins, let's say it notices that it's empty, so it begins to send that signal out through afferent sensory neurons, right? And it's going to that signal is going to travel to the brainstem, and it's going to be integrated here by the CNS. It's going to travel down the spinal cord, and then it's going to be intercepted by a autonomic ganglia, okay, or an autonomic ganglion, and then that autonomic ganglion is going to send motor responses to various other places that that one signal may trigger. So it could trigger digestion processes by the stomach. It could trigger that rumbling of the stomach that you may hear. It could trigger salivation in your mouth to trigger, to try and get you to eat. Any of that. But the source of that is from this autonomic ganglion right here. So yes, the biggest difference is that between the somatic nervous system that we've already discussed and this visceral motor system, the somatic motor division is basically going to be somatic motor neurons innervating skeletal muscle fibers, and that is mostly voluntary. Those are your conscious movements that you make, completely controlled by that motor cortex, that primary motor cortex, and most of this is controlled by the motor neurons directly synapsing onto skeletal muscle fibers, and then they release that neurotransmitter acetylcholine. Well, when you get to the autonomic motor division, these are going to be stimulating smooth muscle cells, cardiac muscle cells, and many glands, okay? And these are going to be involuntary actions. You are not going to have control over this. And what's another thing that's different is these motor neurons do not directly innervate. They do not directly synapse onto their target cells. Instead, what you have are two neurons that are involved. First is the initial efferent neuron, whose cell body resides in the CNS, okay? This is the preganglionic neuron, okay? And the preganglionic neuron will synapse directly onto the cell body of a second neuron called the postganglionic neuron, which is within an autonomic ganglion in the PNS. So then this axon of this postganglionic neuron will then synapse onto the target cell, in which it triggers a change by releasing various neurotransmitters like acetylcholine or norepinephrine. And then these neurotransmitters may either stimulate or inhibit their target cells. Here's a great example. You see indicated here we have the spinal cord right here, and inside of it you have that gray matter of the soma of the axon, and in the somatic system it just travels out and synapses directly onto those skeletal muscle fibers. Well, in the autonomic nervous system you're going to have the same cell body here, but instead of going all the way to its target cell, there's a second step. It synapses onto this postganglionic neuron, and then that carries the signal to its target cell, which is either smooth or cardiac muscle or a gland, okay? And those are, remember, the differences there are somatic is voluntary and autonomic is involuntary. And in the length of these axons depends on which particular branch of the ANS they are in. In the sympathetic nervous system, you'll find that preganglionic neurons axons are very short and postganglionic axons are very long, while the opposite is true in parasympathetic nervous system responses. In the parasympathetic nervous system, you're going to find that preganglionic neuron axons are very long, while postganglionic axons are very short. And here's a great example we see here. This, of course, is still the spinal cord. We see a preganglionic neuron with a very short axon that synapses onto this postganglionic neuron, which its cell body resides inside this ganglion. Remember that. The soma of the postganglionic neuron resides in this ganglion, this autonomic ganglion. And then it has a very long axon that will then synapse onto the target cell, and that is the sympathetic nervous system. The parasympathetic nervous system has very long preganglionic neurons and very short postganglionic neurons. But keep in mind, this location where this soma is for this postganglionic neuron is still the ganglion. It is still the autonomic ganglion. No matter where it is, wherever this second cell body is going to be your autonomic ganglion. And then, of course, that postganglionic neuron synapses onto the target cell. Now, as you saw from that figure, the sympathetic nervous system's neurons, cell bodies, okay, the cell bodies of the sympathetic nervous system often originate near the thoracic and upper lumbar spinal cord. Okay, that preganglionic neuron will originate in the thoracic and upper lumbar spinal cord. And for this reason, the sympathetic nervous system is often called the thoracolumbar division. Okay, and these neurons will synapse first in the sympathetic ganglia, and then these are generally located near the spinal cord, with a few exceptions. And then the postganglionic neurons carry that signal over to the target cell. As I said before, the sympathetic nervous system is usually described as the fight or flight division of the ANS. And this is basically talking about how it prepares the body for emergency situations in which one would either need to fight off an attacker or flee from danger. And this sympathetic nervous system's role is broader than this, however, as it maintains homeostasis whenever the body is engaged in any type of physical work. Okay, and it also mediates the body's visceral responses to emotion. Now, where the preganglionic cell bodies were located within the thoracic and lumbar division of the sympathetic nervous system, of the spinal cord for the sympathetic nervous system, for the parasympathetic nervous system, what you're going to find are these are more located within the brainstem, cranial nerves within the brainstem and the sacral region of the spinal cord. Okay, so where the sympathetic nervous system is called the thoracolumbar division, this parasympathetic nervous system is called the craniosacral division. And what you're going to find is that the cranial nerves will innervate several structures of the head and neck. They'll innervate the thoracic viscera and most of the abdominal viscera, while the sacral nerves are going to innervate structures within the pelvic cavity. And then, as with sympathetic division, okay, these preganglionic neurons synapse first in the parasympathetic ganglia, and then the postganglionic neurons then innervate the target organs. Where these differ is, remember, the length of the axon. So the parasympathetic ganglion is typically located near or within the target organ, as opposed to near the spinal cord. And so that gives that postganglionic neuron a very, very short axon to make the connection. And this parasympathetic nervous system is often called the rest and digest division of the ANS. And this reflects its role in digestion and maintaining the body's homeostasis when at rest. And again, what you see here is a great example for the thoracolumbar division, the sympathetic division. You see these very short preganglionic axons, right? And then very long postganglionic axons to the target organs. And then when you look in the parasympathetic, the craniosacral, you're going to see these very long preganglionic axons and then very short postganglionic axons. And so remember, these ganglions are going to be very near or even within the target organs in many cases. And so again, that difference is sympathetic, parasympathetic. I hope you can, yeah, that's a P. So sympathetic has very short pre, long post. Parasympathetic has very long pre, short post. And then again, another division is thoracolumbar versus craniosacral. Craniosacral. And then thoracolumbar. All right, I hope that makes sense. Now, these don't work independently. What happens is the actions of the parasympathetic nervous system are going to directly antagonize, they're going to go directly against the actions of the sympathetic nervous system. So these guys are headbutting each other this entire time. For an example, the parasympathetic neurons will decrease the rate of your heart rate, whereas the sympathetic neurons increase the heart rate. And so together, these two divisions work to maintain a delicate balance to ensure that homeostasis is preserved at all times. So I want you guys to kind of remember some of this stuff. So I put this in here. This study boost is to remember the difference between pre and post ganglionic neurons. So let's break it down. What is a ganglion? A ganglion is going to be a cluster of neuronal cell bodies. So that is gray matter in the PNS, right? And so if you attach those prefixes pre and post, pre meaning before and post meaning after, then preganglion means that the preganglionic neuron is going to be before the ganglion, and the postganglionic neuron is going to be after the ganglion or within the ganglion. So that is a very important thing to remember. So let's say I'm going to do a little drawing here on the bottom. Let's say this is our spinal cord, and then we have, let's say, a cell body here, and then it goes out to a ganglion not very far out. We'll say this is the ganglion, and then there's another neuron there, and a very long path to, let's say, some smooth muscle. Very artistic, I know. You don't have to praise me too hard, I promise. So this would be an example of what kind of neuron, which division of the ANS is this? And you should know this because I just went over it. So it has a very short preganglionic neuron and a very long postganglionic neuron. I'm sorry, it has a very short preganglionic neuron's axon, and the axon of the postganglionic neuron is very long. So which division is that? It's found in the thorax, in the thoracic region, in the lumbar region. If you said sympathetic, then you're right. It is part of the sympathetic nervous system. This is part of that fight or flight. And then the opposite would be true for a region in the parasympathetic. So let's say you would have an axon there in the sacral region, and then it would go all the way out as far as it could, and then it would synapse in this ganglion here, and then the ganglion would then have a very short axon which would synapse onto the target. Let's just say it's a gland. It would synapse onto the gland. Okay, so you see very long preganglionic axon, very short postganglionic axon. So that is parasympathetic, and that is sympathetic. Please excuse my art. I'm a biology major. I am not an art major. So, sympathetic division of the ANS, fight or flight. That's all I need to know, right? Wrong. Okay, fight or flight grossly oversimplifies this system, because it's not only active during that particular, let's say, a fight or flight situation. It's also active when you experience very strong emotions like fear, excitement, rage, embarrassment. And then it's even active during minor physical activities. In fact, one of the act of standing up requires that sympathetic nervous system. If its neurons don't temporarily initiate changes in your heart rate and blood vessel diameter, your blood pressure will drop quite low due to the effect of gravity on your blood. So if you've ever stood up and you've gotten very lightheaded, you're suffering from a condition called orthostatic hypotension, okay? And this is just a slight failure in your sympathetic nervous system to change your blood pressure as you were supposed to, or as it was supposed to. Now, if you guys remember from that image, you saw many of these sympathetic ganglia are located very closely together along the vertebral column. This gives it a sort of chain-like appearance and has given rise to the name the sympathetic chain ganglia. And so these ganglia extend beyond the thoracic and lumbar spinal cord, and they go as far from the superior cervical ganglion all the way down to the inferior sacral ganglion. And remember, so these preganglionic neurons, these are going to originate in the lateral horns of the thoracic and lumbar spinal cord, and they're going to exit with axons of lower motor neurons through the anterior root. If you guys remember, I talked about all efferent neurons leaving through that anterior root. Well, these are efferent neurons, and they're just originating from the lateral horns. So that's where they differ. The preganglionic axons are going to quickly separate from the spinal nerve anterior ramus to form what is a small nerve called the white, and that just refers to the fact that it's myelinated. So this is going to be the white rami communicantes, and this is going to lead to postganglionic cell bodies in the sympathetic chain ganglion. I have an image here. This should help explain it. So you see this originates in the lateral horn here, and then it travels down and out the anterior horn, out the anterior root, which this is the anterior root, and then it goes and it's going to break away from this spinal nerve, and it's going to form that white ramus communicans here, okay? And then here is where you're going to find the sympathetic chain ganglion. This is just this one chain ganglion, and there is another that's going to be mirrored on this side, so it's going to be the same sort of structure on this side. Now, some preganglionic neurons pass through the sympathetic chain ganglia without synapsing. They go directly onto the cell bodies in different chain ganglia, or they will go onto collateral ganglia, with many of which are located near the target organ, okay? And many collateral ganglia are located near the aorta, and for this reason they are called pre-aortic ganglia. And you're going to find that other collateral ganglia are located near the organs of the abdominopelvic cavity, and so these preganglionic axons synapsing here are part of nerves that are called the splanchnic nerves. And these synapse on ganglia, including the celiac ganglion, the superior mesenteric ganglion, and the inferior mesenteric ganglion. And here we have a great image that shows exactly what I'm saying. You're going to see that these sort of all chain together, one after the other, all the way down to this inferior sacral ganglion. And So the cell bodies are going to be located in here, in that gray matter, and they travel out to the ganglion, and then the ganglion are either going to synapse, like here you see they synapse onto efferent neurons that will innervate the thoracic viscera. You see they go up here to innervate the structures of the head and neck, and then down here they may synapse together into this this aortic structure. And so these are actually going to be the these are going to be the aortic structures up here. These down here are going to be the axons of the splanchnic nerves, where they all go together and they innervate the structures of the abdominopelvic cavity. So there are essentially three ways a preganglionic neuron may synapse with a postganglionic neuron. Okay, the first is the axon will descend or ascend and synapse with a postganglionic neuron in a different chain ganglion. Okay, that's the first. The second is the axon, the preganglionic axon will synapse with a postganglionic neuron in their sympathetic chain ganglion. The third is going to be, let's say this preganglionic axon passes through the chain ganglion and synapses with a postganglionic neuron in a collateral ganglion. So here we should have an image. I'm sorry, the image is after this slide. The postganglionic neuron, okay, this axon is going to run its course and then it's going to innervate the target cells. Now, these postganglionic axons can take the first two pathways to exit the ganglia as small gray rami communicantes. They're called gray because they're unmyelinated, right? These are going to rejoin and travel with spinal nerves until they reach their target cells. And so in contrast, what you're going to see is that postganglionic axons taking the third pathway travel to their target cells directly. And the largest population of postganglionic axons that take this third path are going to be splanchnic nerves. And so let's look at that image, and this should communicate it a little more clearly. So we see the first path. What we're going to do is we're going to follow this one nerve here that starts in the lateral horn and it exits out the anterior root. And then this is that preganglionic axon. So we're following it to the chain, the sympathetic chain. And then that's where we're going to split into our three different synapse methods. So the first, we'll start here. This is going to be the axon descends or ascends and synapses with a postganglionic neuron in a different chain. Okay, so what we see is this axon here travels up and synapses here in this ganglion instead of its parallel ganglion. Okay, so it synapses here, carrying out that signal to the target cell. And so remember, this is still a relatively short axon that that then has a very long postganglionic axon. The second method, that's a 2, I promise. This is going to be the axon synapses with a postganglionic neuron in the sympathetic chain ganglion. Okay, so this is going to be following this same path here, and what we see is that instead of going up or down, it synapses directly in the chain, and then this one carries, this postganglionic axon carries the signal to its target cell here. So it can either go up or down, or what can happen is that this, let's just follow this one down here. For 3, this preganglionic axon will go all the way around, bypassing, in many cases, let's just go this way. What it will do is it will go through the sympathetic chain ganglion and then travel out to a collateral ganglion, which is near the target cell. And this is going to be the only time you find postganglionic axons with short axons here. So yeah, you see the axon passes through the ganglion and synapses with the postganglionic neuron in what is called a collateral ganglion. I hope that has made sense. Sorry if my flow is a little interrupted. I took a break to grade your guys' your tests. So I completely don't remember what I was saying in the last slide. I want you guys to remember that neurons interact with the other neurons, right? And they do so through the release of chemicals called neurotransmitters. And so these neurotransmitters are going to interact with the target cell by binding to a protein-based receptor for that neurotransmitter embedded in the plasma membrane of the target cell. And we're going to see several classes of sympathetic neurotransmitters as we move along through here. So first off, the sympathetic preganglionic axon is going to communicate with the postganglionic neuron by way of using an excitatory synapse and the neurotransmitter acetylcholine. And so this is the same neurotransmitter that is released by somatic motor neurons, as you guys already learned from the muscle tissue chapter. And the unmyelinated axons of the postganglionic neurons will then carry this message from in the form of an action potential to their target cells. So at the synapse with their target cell, the postganglionic axon is going to release one of 3 different neurotransmitters. It could be norepinephrine, epinephrine, or acetylcholine. Epinephrine is often referred to as adrenaline, and norepinephrine is often referred to as noradrenaline. They get their names from the adrenal gland, where these chemicals were first discovered. It turns out that approximately 80% of postganglionic sympathetic neurons are going to release norepinephrine, and then the remainder release either epinephrine or acetylcholine. Now, there are several classes of sympathetic receptors, and it's first important to know that these neurotransmitter receptors that bind to norepinephrine or epinephrine are called adrenergic receptors, and those that bind to acetylcholine are cholinergic receptors. So the first we're going to talk about are cholinergic receptors that bind to acetylcholine. Now there are 2 types of cholinergic receptors. There are the muscarinic receptors, so named because they bind the poison muscarine or muscarine. There are also nicotinic receptors, named so because they bind the poison found in tobacco, that nicotine. Muscarinic receptors are found in the sweat glands on the skin, and nicotinic receptors are located in the membranes of all postganglionic neurons, as well as the cells of the adrenal medulla, which itself consists of modified postganglionic neurons. And so we go back to the adrenergic receptors. These adrenergic receptors are, remember, these are the ones that bind norepinephrine and epinephrine. And there are 2 major types of adrenergic receptors. There are alpha receptors and beta receptors. These receptors are further classified according to their structure of their proteins. Okay, so alpha actually breaks down even further into alpha 1 and alpha 2 receptors. Alpha 1 receptors are going to be located in the plasma membranes of smooth muscle cells of many different organs, including the blood vessels that supply the skin, organs of the GI tract, and the kidneys. These receptors are also found in the erector pili muscles of the dermis, They're in the uterus during pregnancy, and then they're also in certain organs of the genitourinary tract. Alpha-2 receptors, they're going to be a little different than the other adrenergic receptors. We find them in the membrane of preganglionic sympathetic neurons rather than in the peripheral target cells. Okay, remember that normally an action potential in a preganglionic neuron leads to an acetylcholine release, okay, which will then stimulate the postganglionic neuron. However, when norepinephrine will bind to an alpha-2 receptor, the axon terminal is hyperpolarized, slowing or even canceling these action potentials. And as a result, the preganglionic neuron stops stimulating the postganglionic neuron, which can dampen or even shut off the sympathetic response. And this is part of that negative feedback loop that prevents excessive sympathetic activity. You'll find alpha-2 receptors in the plasma membranes of certain sympathetic target cells, including cells of the pancreas and adipose tissue. All right, so here we have an image straight out of your textbook. And so we're going to basically divide this in half. And so here you see alpha-2 receptors with no norepinephrine in them. And then here you see alpha-2 receptors with norepinephrine in them. And so we'll focus on the ones without norepinephrine first. In this case, there is no norepinephrine hyperpolarizing this axon terminal. And so the acetylcholine is able to exit through exocytosis, and it gets transmitted down to the postganglionic neuron easily. This is a sort of normal situation. Now, when these alpha-2 receptors are bound to norepinephrine, what happens is it hyperpolarizes this axon terminal, and it completely prevents any exocytosis. So these are just blocked off. They cannot spit out their acetylcholine to the postganglionic neuron. And so that will reduce activity down the line. And so I just want you guys to know that these are alpha-2 receptors here. These are alpha-2 receptors. And then this one here has norepinephrine in it, and this one does not have norepinephrine in it. So it's important that you guys know that. And remember, there are two different types. There are alpha, and now where there are beta. Beta can also be broken down into further subtypes. Beta 1, beta 2, and beta 3. Beta 1 receptors are going to be located in the plasma membranes of cardiac muscle wall cells, as well as certain cells of the kidney and adipose tissue. Beta 2 receptors are found in the plasma membranes of smooth muscle cells lining the airway passages of the lungs. These are the bronchioles. And they're also found on skeletal muscle fibers, smooth muscle cells of the urinary bladder, and smooth muscle cells lining the blood vessels serving skeletal muscles, liver cells, pancreas cells, as well as some of the cells of the salivary glands. And then beta 3 receptors are located predominantly on the cells of adipose tissue, as well as smooth muscle cells in the walls of the digestive tract. Now, if we're talking about the effects of the sympathetic nervous system on its target cells, the easiest way to understand them is if you understand the system's most basic function. The sympathetic nervous system is entirely designed to ensure survival and maintain homeostasis during times of physical or emotional stress. So think about what happens to your body when you're running a race. Your heart beats faster and harder. You begin to sweat. Your blood pressure rises. There's many other changes. And much of this, the same happens, although to a lesser degree, when you're nervous about a test or excited about an upcoming vacation. Each of these situations will trigger a release of norepinephrine, epinephrine, and acetylcholine from either the pre- or post-ganglionic sympathetic neurons, which will then in turn trigger changes in the cells of target organs and tissues. And you're going to see various effects on different types of tissues. Effects on cardiac muscle cells. As you already know, when you're nervous or exercising, your heart rate and blood pressure rise. This is due to the effect of norepinephrine binding to beta-1 receptors on the cardiac muscle cells, which opens ion channels and increases both the rate and force of contractions. So this heightens not only the amount of blood delivered to tissues, but also blood pressure, which maintains homeostasis during periods of increased activity. Now, the smooth muscle cells of many organs have receptors for norepinephrine as well, and when norepinephrine will bind, it mediates several changes. First, there's constriction of blood vessels, which serves the digestive, urinary, or integumentary systems. So that is your gut, your bladder, and your skin. Now, when norepinephrine will bind to that alpha-1 receptor on these smooth muscle cells, on these blood vessels, these will contract. And so this vasoconstriction, this narrowing of these blood vessels, will decrease the blood flow to those organs and divert that blood to tissues that are temporarily, quote unquote, more important. And so decreasing the blood flow to the urinary and digestive system slows the rate of urine formation, and it slows digestion. For example, let's say you just spotted a bear in the woods and now you're running from it. Digesting the sandwich you just ate can actually wait. Now, this wide-scale vasoconstriction also increases overall blood pressure because it's reduced blood flow to the skin. Reduced blood flow to the skin will explain why people with light-colored skin actually turn as white as a sheet when they're scared, because the blood vessels of their skin constrict and the pinkish hue of the blood under their skin is less visible, causing them to appear more pale. And so all of that is under constriction of blood vessels. There's also the dilation of the bronchioles. So during times of stress, more oxygen needs to be delivered to the body's cells, especially those of the skeletal muscle cells. And so to keep up with that metabolic demand, this norepinephrine's effect on smooth muscle cells lining these bronchioles, when norepinephrine will bind to the beta-2 receptors on these cells, they'll relax. And so this bronchodilation, okay, increases the diameter of the bronchioles, and so more oxygen can be inhaled with each breath. Although the sympathetic nervous system does increase oxygen intake, note that this system has no effect on the rate of ventilation. The rate does tend to increase during exercise and emergency situations, but this is a mechanism dependent of the this is a mechanism independent of the sympathetic nervous system. Okay, more effects. We're continuing on our effects tour. Now, we've already discussed dilation of the bronchioles and constriction of blood vessels serving the skin, digestive systems, and the urinary systems. Now, we're going to find out that the blood vessels serving skeletal and cardiac muscle dilate. Okay? Now, this is the same effect. While norepinephrine constricts many of the body's blood vessels, when it binds to the beta-2 receptors of smooth muscle cells, of blood vessels, in skeletal or cardiac muscle, it causes them to relax. And so this opens up these blood vessels. This is called vasodilation, which increases blood flow and the delivery of oxygen and nutrients to these cells. If this vasodilation only is all that happened, blood pressure would massively decrease. However, it occurs at the same time as the vasoconstriction of nearly every other blood vessel in the body. So the effect of skeletal and cardiac muscle vasodilation on blood pressure is actually relatively minimal. And then there's also contraction of the urinary and digestive sphincters. The binding of this norepinephrine to the smooth muscle cells of the sphincters of both urinary and digestive systems causes them to contract. This makes emptying the bowel and bladder during times of exercise, stress, or emergency much more difficult. So more effects. There's relaxation of the smooth muscle by the digestive tract. And so, although stimulation by sympathetic nervous system causes this smooth muscle to contract in the digestive system, when norepinephrine binds to beta-3 receptors, it leads to the relaxation of the remainder of the smooth muscle. And so this actually decreases the movement of food through the digestive system, and it overall slows the digestive process down. Your pupils will also dilate. The dilator pupillae muscles are innervated by sympathetic nervous system neurons. And when norepinephrine binds to the alpha-1 receptors on these muscle cells, it causes them to contract, which dilates the pupils, allowing more light to enter the eyes. The blood vessels serving most exocrine glands will constrict. The sympathetic nervous system almost universally decreases secretion from exocrine glands apart from sweat glands through norepinephrine binding to beta-2 receptors. This affects most exocrine glands of the digestive system, including the salivary glands around your mouth and the cells that secrete digestive products. This effect on salivary glands is why your mouth can often be dry when you're nervous. And so the decreased secretion is generally due to a reduced blood flow to these glands rather than a direct effect on the gland itself. And then during times of sympathetic nervous system activation, nearly all cells, in particular skeletal muscle cells, consume way more ATP as their metabolic rates increase. And so to ensure that muscle cells have a steady supply of fuel with which to make ATP, norepinephrine brings about three main effects. It binds to beta-3 receptors in adipocytes and triggers the breakdown of lipids. And then this will actually release free fatty acids into the bloodstream. Next, it will bind to beta-2 receptors on cells of the liver, and this will trigger the release of glucose from glycogen, as well as the synthesis of glucose from other precursors. And then it can also bind to beta-2 receptors on cells of the pancreas and trigger the release of a hormone called glucagon, and this increases the concentration of glucose in the blood. Before I actually talk about the sweat glands, I want to finish talking about the digestive system. Be careful that you don't confuse metabolic rate with digestion, okay? Metabolic rate is going to be the rate at which cells consume ATP, and this increases during sympathetic activity, while digestion is the breakdown of food into nutrients that can be absorbed by blood, and those processes decrease when sympathetic activity increases. So I want you guys to be able to separate digestion from metabolic rate. Now, as I said, there are the sympathetic nervous system does affect sweating differently. When you're nervous or exercising, you sweat. This useful but often annoying attempt to help the body maintain temperature homeostasis is due to that post-ganglionic release of acetylcholine into cells of sweat glands of the skin. This acetylcholine binds to these muscarinic receptors and increases their secretions. This is another negative feedback loop that controls body temperature, which goes all the way back to the feedback loop's core principle that you guys have talked about. So here you see the various breakdowns, as well as I want you guys to remember this is that sympathetic chain over here, and it shows you various regions that innervate the different glands or organs. And so you see there are the targets here, the neurotransmitter here, the receptor, okay, so that is an alpha-1, you got beta-1, beta-2, beta-1, and then you have the main effect of sympathetic stimulation, I'm not going to go through each of these individually, but it is important that you guys know what the effects of these sympathetic stimulations are. So I'll leave this up here just for a second for you guys to pause and get a good look at. Don't forget, this is also in your textbook. All right, I'm moving on to the next slide now. Now, these adrenal glands that we talk about that secrete norepinephrine and epinephrine, now, they sit on top of each kidney, okay? And the adrenal medulla is the internal part of these glands. And what is unique is that it is contacted directly by preganglionic sympathetic neurons. This is because the adrenal medulla is not like the rest of the adrenal gland. Instead of the glandar epithelium, that it actually has modified sympathetic postganglionic neurons. So essentially, each adrenal medulla is functionally a ganglion. Now then, as I said, this preganglionic neuron is going to secrete acetylcholine into the adrenal medulla, okay? This adrenal medulla is going to have various cells that accept this acetylcholine, and then they begin producing norepinephrine and epinephrine in massive numbers. So once that begins happening, this norepinephrine and epinephrine then travels into the bloodstream as a hormone, as that long distance chemical messenger. Do remember that this acetylcholine binds to the nicotinic receptors of adrenal medulla cells. That is important. Now, the ratio of chemicals in this adrenal medulla that it produces is generally the opposite of that of the autonomic nervous system. The adrenal medulla will produce about 80% epinephrine and 20% norepinephrine. This additional epinephrine and norepinephrine is useful in many ways in prolonging the duration of the sympathetic nervous system's effects. The effects of neurotransmitters released by these postganglionic neurons will typically only last a few seconds, whereas in the blood, the effects of these chemicals continue for several minutes. A second benefit is that epinephrine and norepinephrine in the blood can reach cells that are not innervated by the sympathetic neurons. So these normal neurotransmitters, epinephrine and norepinephrine, when they act as hormones, they're able to reach a wider, quote-unquote, a wider audience of cells. And so this allows that sympathetic nervous system to affect the entire body without innervating every single cell. And then finally, the adrenal medulla acts as a sort of backup for the sympathetic nervous system. Even if the pathways connecting the system with any of its target organs are disrupted, it can still affect these organs through the adrenal medulla, through the production of norepinephrine and epinephrine as hormones instead of neurotransmitters. Now, what effects does the sympathetic nervous system have on other cells? Well, the sympathetic nervous system influences many other processes through its effects on its target cells. For instance, this system enhances mental alertness, and this increases activity in the association areas of the neocortex. As well as this system also increases the blood's tendency to clot, which can be helpful if a person is injured during one of those fight or flight situations. Also, it can temporarily elevate the tension generated by skeletal muscle cells during a muscle contraction, which is why you've ever heard of those people like a mother with her baby trapped under a car. She's able to lift that car under the influence of an epinephrine rush, an adrenaline rush. Furthermore, these sympathetic neurons trigger the contractions of the erector pili muscles, which can produce those goosebumps when you feel a strong emotional reaction. And then finally, in both male and female ejaculation, these neurons are also the source, the cause of the ejaculation via effects on smooth muscle cells of the reproductive ducts, or the lubrication ducts in the female body. Now, because there are very specific subtypes of these sympathetic nervous system receptors, this allows researchers to design drugs that are very specific for one type of receptor and thus certain organs. You're going to find that many different disease states like asthma and hypertension are caused by these sympathetic nervous system blockers or enhancers. And so they basically work in one of two ways. They are either antagonists blocking the receptor, preventing norepinephrine from binding to it, or they are agonists and they bind to that receptor, mimicking the effects of norepinephrine. And so there are several common types of drugs that bind to these sympathetic receptors, and they are alpha-1 blockers. These are going to be antagonists binding to alpha-1 receptors of smooth muscle cells in the blood vessels. Now, they block norepinephrine, preventing the blood vessels from constricting. Okay, and so this is an effect that lowers blood pressure and is useful in treating hypertension or high blood pressure. Certain alpha-1 blockers also cause the relaxation of smooth muscle cells in the prostate gland, and this can be useful for treating prostatic hyperplasia. So, some examples include tamsulosin, doxazosin, and terazosin. If you're going into nursing, I'm sure you'll learn all about these. You also have alpha-2 agonists, and these are going to bind to the presynaptic alpha-2 receptors and activate them. So this decreases the output of both preganglionic and postganglionic sympathetic neurons. These drugs may be used to treat hypertension and other conditions like opiate withdrawal. So these are going to be clonidine, guanfacine, tizanidine, phenylephrine. So yeah, these are going to be alpha-2 agonists. Then you have beta blockers, and beta blockers are antagonists that bind to beta-1 receptors on the heart and decrease its rate and force of contraction. These drugs are widely used in the treatment of hypertension as well as other diseases of the cardiovascular system. And then you've got beta-2 agonists. And beta-2 agonists bind to beta-2 receptors on the smooth muscle of the bronchioles, causing bronchodilation. And these are essentially asthma drugs like albuterol. And the examples of beta blockers were metoprolol, propranolol, and carvedilol. So these are going to be very, very common drugs if you go into the nursing practice that you'll get to know. And now you know the action behind them. Now, as I previously told you guys, one effect of the sympathetic nervous system is that it increases metabolic rate. And so this increases the rate at which ATP is produced and consumed. Now, this has led to some shady dietary supplement manufacturers creating products intended to result in weight loss by capitalizing on this effect. And these products are generally sold as topical creams that the user will, quote, rub over problem areas with excessive adipose tissue. And it turns out that in many of these creams there is a chemical called yohimbine, and this is the active ingredient derived from a plant called the yohimbe plant. And manufacturers claim that yohimbine binds to beta-3 receptors on adipocytes and triggers the breakdown of lipids. At best, this is a misleading statement because yohimbine actually blocks alpha 1 receptors in blood vessels and alpha 2 receptors in the spinal cord. So it has a dual effect. It causes vasodilation while also increasing the activity of the sympathetic neurons. This higher sympathetic activity can briefly increase metabolic rate. However, it dangerously elevates the heart rate and can cause seizures, high blood pressure, kidney failure, and ultimately lead to insomnia and panic attacks. Fortunately, yohimbine is not actually absorbed through the epidermis in any significant amount, so it never really reaches the blood vessels in the dermis or hypodermis, and this limits the amount of harm it can do to your body. However, it can still harm your wallet. So what is it you guys learned out of this chat, out of this particular PowerPoint? Well, we did a brief overview of the autonomic nervous system. You learned the differences between the somatic and the autonomic motor systems. Then you learned the different branches of the autonomic nervous system, the sympathetic versus the parasympathetic systems. We've delved deeply into the sympathetic nervous system, talking both about it, its neurotransmitters, and the effects of the sympathetic nervous system. And so in the next one, we'll continue on through chapter 14, and we'll continue on through parasympathetic nervous system and homeostasis and how all of that works together with the CNS. I will see you guys in the next one.